Swayam Prabha. Digital India. Educated India.
Welcome back. Today we will start the topic on protein energy landscape. Okay. So, I am planning to discuss the concept on protein folding, because the energy landscape is very much related to protein folding, and you remember that we started to discuss about this protein folding. The definition I gave on the last week. Now, we also discussed the Monte Carlo simulation technique, but there are some problem with the Monte Carlo simulation technique. And although we demonstrated that for the integration of an equation or say for computing the value of the pi, the Monte Carlo simulation technique is perfect. But for our purpose, I mean for protein folding problem, this Monte Carlo simulation technique even along with the Metropolis criteria may create some problem. So, the problem actually is triggered by the nature of the protein energy function. Although we designed one energy function, but you remember that I mentioned when I say doing the integration of one function, say integration of f(x) dx, sometimes it is difficult to actually have that effect. So, we approximate to gx. And even if we approximate to gx, then we will find that there is some nature. Because of that nature, actually it might be a problem to run only the Monte Carlo simulation technique, even if there is Metropolis criteria. So, those things we will discuss in today's lecture. So, that is why the protein folding, protein energy and the protein energy landscape, what is it? So, those things we will discuss today, and the keywords, that is why I selected protein folding and energy landscape. So, let us recap with the definition of the protein folding problem. It says that given one protein sequence as an input, which we can also call as protein primary structure, you need to determine the 3-dimensional structure of it, or you need to determine the atomic level resolution or the atomic model corresponding to that input sequence, without the need for experimental verification or validation or testing. Okay. So, that is about the protein folding problem, and here the diagram indicates that the same diagram that we discussed on the last week. So, FASTA is my input. Since it is a sequence, so I am mentioning it as a FASTA, but actually you can consider that this FASTA is nothing but the protein sequence, that also that you can also see. It is a single letter code. So, M, the first one indicates methionine, N the second one indicates asparagine, L the third, fourth, fifth one is indicating the leucine, T indicates the threonine, like that way it is. Now, if there exists one black box, that is basically the computational technique or a software tool which will take this FASTA as an input and will give me this 3-dimensional model as an output. So, that is my requirement, that is the protein folding problem. However, when we are say designing this black box or the software tool which will take one sequence as an input and will give me one structure as an output, so there may be some possibilities, it may not be the correct one. And if I go back to the biology and or say chemical biology and try to see that what is the behavior of a protein when say actually in vivo or in cell it is behaving, then we will see some interesting features of that particular protein. So, that features we will discuss first. So, first thing is that given one protein sequence, it is supposed to fold into a particular structure, and that particular structure will have one function. So, for example, you can consider that once a sequence is given and it will fold to this structure and it will have one function, but not necessarily it will always fold to that one. There may be several reasons, say environmental pressure or say mutations. Mutation means that some point has changed, because of that one it may not take this particular shape or structure. Now, you know that if one particular shape or structure is there, then based upon that one function will be determined. So, for example, if I assume that okay, so the basic material is always a steel. Now, out of that steel I can say make one shape like this. I can have something like this, I can have something like this. So, if you assume that say this is my fork, this is my spoon, then this has one function, this has one function, and there is kind of a correspondence between the structure and function. Say instead of say forming the spoon, if I form a fork, so that is also a structure that you see, that say I am taking say melted steel, and instead of making a spoon I am basically make one fork. Then all the functions of a spoon, say for example say drinking soup, so that property will not be satisfied by this fork, right. So, if that is the situation, then during this sequence to structure folding process, if something goes bad or goes wrong, then what will happen, that because of this mutation, environment pressure, or say change of pH, etc., that we will discuss. Okay. Then if this particular shape is not assumed, then one particular function will be absent. I believe you all know that inside the ribosome, actually from DNA to miRNA and then to protein, the transcription translation process will take place, and when say miRNA is basically translating to protein, then it will take some shape that we will call as a fold, and as if that this will move from this sequence to this structure. Now, if because of anything this particular structure is not assumed, and say it is assuming one structure like this, then clearly you can see that the blue structure and the red structure is different, also their functionality will be different. That is why you say, if I assume that this particular protein sequence is a protein sequence of a human body, then if instead of blue structure, which is actually native structure, native means that it is the functional form and it has one particular function, instead of that one, if it assumes red structure, then according to that red structure it will have some function, but that function is not same as the blue function, whereas your body is waiting for the blue structure, and corresponding to that blue structure there is a function, and that function will be absent in your body. So, you may lead to some situation which can be a disease condition. So, that may happen if a protein misses to fold or misses to assume its correct folding structure. Okay. Related to that, there are three concepts: folding, unfolding, misfolding. So, when a particular sequence assumes a correct structure, When I say correct, which means that it is known that this sequence is supposed to take this particular structure, and this process I am talking in the context of the in vivo, which means inside the cell. I am not talking about developing one algorithm who is actually folding. So, in biology or in chemical biology, when it assumes one particular structure corresponding to one sequence, when it assumes that particular structure, then it is called as a folding. However, in the process, if it happens that instead of blue structure it is taking the red structure, because of that one it folds, but that is not a correct fold. So, I will call that as a misfolding. Now, you will be surprised to know that in our body there is a mechanism through which it can senses that whether a protein is assuming a correct structure or not most of the time. And the moment it will recognize that okay, probably it is not assuming the correct structure, then it will allow to misfold, I mean open that folding which was leading to some incorrect folding, and then allow it to fold it again. Now, if for the second chance also it will not able to fold correctly, then definitely it is going to a misfolding state. Okay. And unfolding indicates that when a sequence is not in the folded form. Okay. So, regarding this few features we are going to discuss. The correct 3-dimensional structure is essential to function, although some parts of functional proteins may remain unfolded. So, this is one unique situation which can be found in protein. Say it is assuming one structure like this. Now, you see that this region is folded, but this part is basically not folded. That may possible, okay? But the function will be then corresponding to this one. Okay. Next, failure to fold into native structure generally produces inactive proteins, but in some instances misfolded proteins have modified or toxic functionality. So, when it will fail to fold to native structure, again native means that it has a specific function to do, and for that specific function it should assume one particular structure. And if that particular structure it cannot able to assume or it cannot fold to that particular structure, then what will happen that it will assume another structure. Now, that particular structure may be completely inactive, do not have any function at all, or it may have some function. Now, that function mostly be not good for our body, that is why it will be with the toxic functionality, okay, which is unwanted, not required for our body. Next, several neurodegenerative and other diseases are believed to result from the accumulation of amyloid fibrils formed by misfolded proteins. So, when there is a misfolding, because of that one, that amyloid fibrils are formed. This particular amyloid fibril actually is more stable, fibrils are more stable compared to the native state from stability point of view. But you know that although we are talking about stability of one protein molecule, but of course, the functionality is also related to that stability. If we reach to some situation where it is of utmost stable, but there is no function, then it is useless. So, something like this amyloid fibril, it will reach to some region where the energy is even less compared to the native state, but it do not have any function. That is why, make a note of this, that is why computationally when we are designing some software tool, then definitely we will try to optimize our energy function, or we will try to reach to the minimum energy state or stable energy state corresponding to one protein structure that we are modeling, but that must have one function. If we reach to some situation so that it do not have any function, then that is useless. Then the question may come in your mind that how do I discriminate that whether it will have a function or not. So, that thing you can fix after your modeling, you can basically do some corrections or some cross checking. Cross checking means whether that is assuming a particular structure following that Ramachandran plot, or say whether it is having some fold or not. So, those things you can check, and then perhaps you can say whether it is a good modeling or not. Okay. Many allergies are caused by incorrect folding of some proteins, which means incorrect folding means that that protein is supposed to take some structure and have some function, but it folded incorrectly, and because of that incorrect folding, two situations occurs. One is that you are basically starving for the correctly folded protein, or you are starving for some known or essential protein, because that particular fold it is not assuming, and you are basically generating some incorrectly folded protein whose function is not required for your body. So, unwanted thing mostly leads to some allergy. For the immune system does not produce antibodies for certain protein structures. So, if it is say misfolded or are taking some wrong shape, then it is not possible for say human body to identify that it is misfolded. So, throw it out of the body like it used to do for say foreign bodies. So, when foreign body enters to our body, then our immune system identifies that it is a foreign body. So, throw it out of the body, it may create some problem in our body. Again, some disease, say viral disease or other disease, when they sets in, then they basically cripples the immune system first, and then immune system will not able to track or detect that particular foreign body, and that way they progresses and stays in our body. So, that is one issue, different one, but in this context what I am trying to say, inside the body no foreign body is coming, inside the body the protein instead of taking its native state, it is actually taking some incorrect state, and because of that incorrect state it is unwanted in our body. Sometimes our immune system may identify, okay, this is not required, from where it is coming, okay, I do not care from where it is coming, since it is not required, so throw it out of our body. Sometimes it is possible, but not always. Okay. So, when it is not possible, then it leads to some disease or say some allergy condition. So, here is the schematic diagram for folding, unfolding and misfolding. You see this is one sequence. So, each circle indicates say one amino acid, but you should remember that basically it is not complete, it is extending in both the ways. So, N terminus and C terminus. So, only a small part I have drawn, but there is a long portion of it. Okay. So, one situation that this is unfolded and it will take a properly folded step, that you can see. Here this spiral indicates the helix. That is one regular secondary structure. Okay. Now, it has some shape, or because of some factor it may take some toxic protein clump. Now, you see that this shape is kind of irregular in nature. So, that spirality etc. I cannot able to see here. Now, this toxic protein clump definitely is unwanted in our body and may lead to some disease condition. Okay, may lead to. Sometimes our immune system may identify that this is unwanted and so throw it out of our body. If it is, then it is fine. If it is not, then it may create some problem. Now, this protein, toxic protein clump may occur because of several reasons. Okay. One situation may be that some amino acid during the evolution process or translation process inside our body is changed because of that change it may happen. During the folding process, the others or the environment which is supposed to allow it to assume its correct folded structure, like this green one, is basically not able to support it properly, and because of that one it is going to the red state. Here it is toxin protein clump. Now, who are the protein driving forces? So, first one, although its individual contribution is very small, that is the hydrophobic side chain. So, hydrophobicity. Okay. So, in a protein, say if I assume this is my unfolded state. Okay. So, let us assume that there are some these are highlighted parts are basically say hydrophilic, rest of the parts are hydrophobic. Then if you assume that okay, this particular unfolded protein or partially folded protein, not completed and is not in a straight string form, okay, if I put it into an water, that is my solvent, then it is going to take some shape like okay. So, here this corresponds to say this region, this region corresponds to this region, this region corresponds to this region, and apart from that you see that there are some regions say like this region, okay, which is basically this sum of this part, and this red region is say hydrophobic. They are also exposed to water, but you have to understand that it might be, but the aim is that optimizing the fold in such a way that the contribution of the hydrophobic being inside the core or away from the water is more compared to that hydrophobic will be on the surface. So, hydrophobic, so this amino acids I am classifying into two groups as of now, hydrophobic and hydrophilic. So, this is water loving and this is just the opposite, this hydrophobic. Now, inside this hydrophilic there are variations like charged, then let me go to some press page, right. Charge, polar. Okay. Now, there are some hydrophobic residues and hydrophilic residues. So, hydrophilic loves water molecules. So, it will try to be on the surface, but hydrophobic do not like water molecules. So, examples here I can give as a leucine, lysine, isoleucine, okay. Then say what else, to some extent you can consider. So, TRP, TRP means W, sorry. Okay. So, these are some of the hydrophobic residues. So, there are some hydrophobic scale through which you can measure and you can identify. But the point is that that is, although the contribution of that hydrophobic residue, individual contribution is basically less compared to say charge interaction or say disulfide bond interaction, but the collective or if I take the summation over the individual contributions, then if I take the individual contributions, then basically it will be very high. Okay. So, going back to that slide actually, so minimizing hydrophobic side chain exposed to water is one. Next, solvent, water or lipid bilayer. Now, the point is if it is water, then what will happen, hydrophobic will be at the core and hydrophilic will be on the surface. If it is lipid bilayer, the nature will be opposite. So, you see that based upon what is the solvent, the nature of the folded state will change, and that way you may think and you can think correctly that one particular amino acid sequence based upon what is the solvent may assume different folded structure, and accordingly they have multiple functions. Okay. Concentration of salt. So, salt will give you more ions in the solution, because salt, the simple salt you can assume is NaCl. So, it will contribute Na and Cl. Okay. Now, all the charged molecule, who are the charged molecule? Sorry, charged amino acid. So, one is aspartic acid, glutamic acid, arginine and then K. Okay. So, these are basic and these are acidic. Sometimes H or histidine is also considered as basic. Now, if the concentration of the salt in the environment increases, then whether percentage of plus increases or basic part increases, ion increases, or the acidic ion increases, based upon that one who will be on the surface will be determined. Okay. So, the concentration of the salt will also matter. Then pH, pH and concentration of salt is to some extent related with each other. Okay. So, water has some neutral pH. If you go in one side you will get base, another side you will get acid. Then temperature. Okay. So, each atom has some say B factor, and based upon that one it vibrates. Now, if temperature increases, then the folded state will also change. Possible presence of cofactor or molecular chaperones which helps to take a particular fold. Say in presence of that particular cofactor or molecular chaperone, the fold may be different compared to in absence of that molecular chaperone or cofactor. Intramolecular hydrogen bonds that will make a difference. Again, this particular hydrogen bond or the salt bridge, okay, hydrogen bond, salt bridge, disulfide bond, so these are called as the ionic bond. Okay. So, this kind, so disulfide will always occur between two cysteine or where there is a sulfur, okay. But salt bridge will form between say one acid, another basic. So, acid I mentioned that aspartic acid, glutamic acid, base indicates that arginine or lysine. Okay. So, between this actually there will be salt bridge, and hydrogen bond can be with the main chain, with the side chain. Whenever there is a hydrogen donor and then hydrogen acceptor, then there can be a hydrogen bond. So, the complete sum of this effect will also be a driving force for protein folding process. Van der Waals interaction, okay. So, this is also called as the LJ potential or 612 potential, because there is a 6 and 12 part. So, the equation says that A divided by r to the power 6 r to the power 12 minus B divided by r to the power 6. Okay. So, that part is also there. So, those are the driving force and many other, this electrostatic interaction that I have mentioned as the ionic one, okay, and many more are there. But these are more or less enough for us when we will formulate it to some energy function. Okay. So, these are in a nutshell the driving forces behind the protein folding. So, when we will say design one protein folding software or tool, then in order to design the energy function, then we have to keep all those things in our mind. Okay. Now, I will conclude by mentioning two protein folding models. One is the diffusion collision model, another is the nucleation condensation model. So, folding often begins co-translationally, so that the N terminus of the protein begins to fold while the C terminal portion of the protein is still being synthesized by the ribosome. The diffusion collision model, in which a nucleus is formed, then the secondary structure is formed, and finally, these secondary structures are collided together and packed tightly together. Whereas, for nucleation condensation model, in which the secondary and tertiary structure of the protein are made at the same time. Okay. So, that is it. Now, we will continue to the next lecture, the rest of the part. Thank you.
പെൺകുട്ടികളെ പഠിപ്പിക്കുന്നതിന്റെ ഉദ്ദേശം എനിക്ക് മനസ്സിലാവുന്നില്ല.
എന്ത് പറ്റി മുത്തശ്ശി? ഞാൻ പഠിക്കുന്ന അടുത്ത ആഴ്ചയുള്ള ഒരു ഡിബേറ്റ് കോമ്പറ്റീഷന് വേണ്ടിയാണ്.
ഓ, അതായത് നിന്നെ സ്കൂളിൽ മാത്രം അയച്ചാൽ പോരാ. ഇപ്പോൾ നമ്മൾ പെൺകുട്ടികളെ പഠിപ്പിക്കുന്നത് തറുതല പറയാനാണ്. ഞങ്ങളുടെ കാലം ഇതിനേക്കാളൊക്കെ എത്ര നല്ലതായിരുന്നു. പെൺകുട്ടികളെ നേരത്തെ തന്നെ വിടും. പിന്നെ അവർ പാചകവും വീട് വൃത്തിയാക്കലും ഒക്കെയായി സമയം ചെലവഴിക്കും. ദിവ്യയ്ക്ക് വേണ്ടി നമ്മൾ ഒരു പയ്യനെ കണ്ടുപിടിക്കണം.
അവളിപ്പോ ഒരു കുട്ടിയല്ലേ അമ്മേ. എന്റെ മക്കൾക്ക് എത്രത്തോളം പഠിക്കണോ അതുവരെ ഞാൻ അവരെ പഠിപ്പിക്കാൻ ആഗ്രഹിക്കുന്നു.
കൂടുതൽ പഠിച്ചാൽ അവൾക്കൊരു ചെറുക്കനെ കിട്ടാൻ വലിയ ബുദ്ധിമുട്ടായി തീരും.
ഇല്ല അമ്മേ, ദിവ്യയ്ക്ക് പഠിപ്പുണ്ടായാൽ അവൾക്ക് സ്വന്തമായൊരു പ്രൊഫഷൻ തിരഞ്ഞെടുക്കാം. അപ്പോ അവൾക്കൊരു ഡോക്ടറോ ടീച്ചറോ എന്തോ ആവാൻ സാധിക്കും അമ്മേ. അമ്മയ്ക്ക് അറിയുമോ, ഈ കാലത്ത് ചില രാജ്യങ്ങളിൽ സ്ത്രീകൾ പ്രസിഡന്റും പ്രധാനമന്ത്രിയും ഒക്കെ ആകുന്നുണ്ട്. അതുമല്ല, നമ്മുടെ സമൂഹം കുറേശ്ശെയായി മാറിക്കൊണ്ടിരിക്കുകയാണ്. ഇന്നത്തെ കാലത്ത് ആളുകൾ വിദ്യാഭ്യാസമുള്ള പങ്കാളിയാണ് ആഗ്രഹിക്കുന്നത്. ഹേയ് ദിവ്യ, ഞാൻ ചെയ്തോളാം. ദയവായി നീ പോയി പഠിക്ക്. മുത്തശ്ശി പറഞ്ഞതൊന്നും നീ കാര്യമായി എടുക്കണ്ട. അവർക്ക് അവരുടേതായ പഠിത്തം പൂർത്തിയാക്കാൻ കഴിഞ്ഞില്ല. പരമ്പരാഗത ചിന്താഗതിക്കാരിയാണെങ്കിലും നിന്റെ കാര്യത്തിൽ അവർക്ക് വലിയ ശ്രദ്ധയുണ്ട്.
അമ്മയ്ക്കറിയോ, ഞാൻ പഠിക്കുന്നതിനും വീട്ടിൽ സഹായിക്കാത്തതിനും മുത്തശ്ശി കുറ്റപ്പെടുത്തുമ്പോൾ എനിക്ക് വേദന തോന്നാറുണ്ട്.
ദിവ്യ, നിരാശപ്പെടാതിരിക്കൂ. നിന്നെ പിന്തുണയ്ക്കാൻ ഞാൻ എപ്പോഴും ഇവിടെയുണ്ട്. വീട്ടുജോലി എല്ലാം ഞാൻ ചെയ്തോളാം.
നന്ദി അമ്മേ. ഒരു ദിവസം അമ്മ എന്നെ ഓർത്ത് അഭിമാനിക്കും. നിനക്കറിയാമോ ആര്യൻ, പെൺകുട്ടികൾ ജനിക്കുന്ന കഷ്ടപ്പെടാനും വീട്ടുജോലികൾ ചെയ്യാനും വേണ്ടി മാത്രമാണെന്ന് എനിക്ക് ചിലപ്പോൾ തോന്നാറുണ്ട്.
നീ എന്താ അങ്ങനെ പറയുന്നത് ദിവ്യ?
ഞാൻ പഠിക്കുന്നതിൽ മുത്തശ്ശിക്ക് താല്പര്യമില്ല. എന്നെ കെട്ടിച്ചുവിടാനാണ് ആഗ്രഹിക്കുന്നത്. പെൺകുട്ടികൾക്ക് ആഗ്രഹമൊന്നും പാടില്ലെന്നാണ് അവർ കരുതുന്നത്.
ദിവ്യ, സമ്മർദ്ദത്തിൽ അകപ്പെടരുത്. വരൂ, നമുക്ക് നമ്മുടെ ക്ലാസ് റൂമിലേക്ക് പോകാം. ദിവ്യ, നമ്മുടെ ഭരണഘടന പോലും 18 വയസ്സിൽ താഴെയുള്ള പെൺകുട്ടികളുടെ വിവാഹം അനുവദിക്കുന്നില്ല.
അത് തന്നെയാണ് എന്റെ അമ്മയും എന്നോട് പറഞ്ഞത്. അമ്മ എന്നെ ഡിബേറ്റ് കോമ്പറ്റീഷനിൽ പങ്കെടുക്കാൻ പ്രോത്സാഹിപ്പിച്ചുകൊണ്ടിരിക്കുകയാണ്.
ഒരു മിടുക്കി കുട്ടിയായ നീ തീർച്ചയായും അതിൽ പങ്കെടുക്കണം. പരമ്പരാഗതമായി നമ്മുടെ സമൂഹം പെൺകുട്ടികൾ ഉന്നത വിദ്യാഭ്യാസം നേടുന്നത് വിലക്കാറുണ്ട്. എന്നാൽ പഠിത്തത്തിൽ നിന്നെ നന്നായി പ്രോത്സാഹിപ്പിക്കുന്ന ഒരമ്മയുള്ള നീ ഭാഗ്യവതിയാണ്.
നന്ദി ആര്യൻ.
ദിവ്യ, ഹലോ. ഇന്നത്തെ ദിവസം നിനക്ക് എങ്ങനെ തോന്നുന്നു?
ഇന്നലെ എനിക്ക് പ്രചോദനം നൽകിയതിന് നന്ദി. സമാധാനത്തോടെ പ്രാക്ടീസ് ചെയ്യാൻ അതെന്നെ ശരിക്കും സഹായിച്ചു.
അങ്ങനെ കേട്ടതിൽ സന്തോഷമുണ്ട്. കോമ്പറ്റീഷന് എല്ലാ ആശംസകളും.
താങ്ക്സ് ആര്യൻ.
ഡിസ്ട്രിക്ട് ഡിബേറ്റ് കോമ്പറ്റീഷനിൽ ഈ വർഷത്തെ വിജയിയാണ് ദിവ്യ. നല്ല വിദ്യാഭ്യാസം കാരണം ദിവ്യയ്ക്ക് നല്ല പ്രബുദ്ധതയുണ്ടായി. അവൾക്ക് ആത്മവിശ്വാസത്തോടുകൂടി സംസാരിക്കാൻ കഴിഞ്ഞു. ദിവ്യ, ഞങ്ങൾ കുട്ടിക്ക് 3 വർഷത്തേക്ക് സ്കോളർഷിപ്പും ഒരു ലാപ്ടോപ്പും തരുന്നു. ആൺകുട്ടികളുടെയും പെൺകുട്ടികളുടെയും വിദ്യാഭ്യാസത്തിൽ ഇൻവെസ്റ്റ് ചെയ്യേണ്ടത് ആവശ്യമാണ്. എല്ലാ ലിംഗത്തിൽപ്പെട്ടവർക്കും തുല്യ അവസരങ്ങൾ നൽകേണ്ടത് ആവശ്യമാണ്. ഇന്ന് ഇതിൽ പങ്കെടുത്ത എല്ലാവർക്കും നന്ദി. അഭിനന്ദനങ്ങൾ ദിവ്യ.
നന്ദി മുത്തശ്ശി.
എനിക്ക് അത്ര വിദ്യാഭ്യാസമൊന്നുമില്ല, പക്ഷെ വിദ്യാഭ്യാസത്തിന്റെ മഹത്വം ഇപ്പൊ ഞാൻ മനസ്സിലാക്കുന്നു. എനിക്ക് നിന്നെ ഓർത്ത് അഭിമാനമുണ്ട്.
ഓരോ വ്യക്തിയുടെയും മൊത്തത്തിലുള്ള ശാരീരികവും വൈകാരികവുമായ സന്തോഷത്തിന് പരസ്പര ബന്ധങ്ങൾ പ്രധാനമാണ്. ഒരാൾ മറ്റൊരാളുടെ വികാരങ്ങൾ മനസ്സിലാക്കുന്നതിന് നിരീക്ഷിക്കുകയും ശ്രദ്ധിക്കുകയും ചോദിക്കുകയും ചെയ്യേണ്ടത് ആവശ്യമാണ്. സുഹൃത്തുക്കൾ, സഹോദരങ്ങൾ, മാതാപിതാക്കൾ അല്ലെങ്കിൽ അധ്യാപകർ എന്നിങ്ങനെ നമുക്ക് ചുറ്റുമുള്ള ആളുകളുമായി നല്ല ബന്ധം സ്ഥാപിക്കാൻ ഈ കഴിവുകൾ നമ്മെ പ്രാപ്തരാക്കുന്നു. മറ്റുള്ളവരുടെ വിചാരങ്ങളും വികാരങ്ങളും നാം മനസ്സിലാക്കുമ്പോൾ അവരുടെ ആഗ്രഹങ്ങളോട് നാം കൂടുതൽ സംവേദനക്ഷമത പുലർത്തുന്നു. അത് നമ്മുടെ ബന്ധങ്ങളെ ശക്തിപ്പെടുത്തുന്നു.
Swayam Prabha. Digital India. Educated India.
Welcome back. So we started to discuss regarding the protein energy landscape, and at the beginning started with the protein folding problem. I mentioned the forces which are responsible probably for the protein folding, and during the process, even in vivo, there is a possibility that the protein may assume some wrong folded state. Wrong in the sense that corresponding to each protein sequence, it is supposed to assume one particular structure, and there is a one-to-one correspondence between the structure and the function. But it may happen that that particular structure is not assumed because of some reason during the protein folding process in cell. And if it is not, then what will happen, that it may lead to some misfolded state which may be say may create some disease or some allergy. It may be toxic to our body, or our immune system can able to identify that and throw it away. Whatever it may be, when one protein sequence assumes to take one particular folded state, then we are definitely missing its functionality in our body. So, whatever function it is. Now, that thing we concluded on the last slide regarding 2 different folding models that exist. One is the belief is the diffusion collision model, another is the nucleation condensation model. Okay. Now, here we will continue with the protein energy landscape, and we will start discussing regarding what is the limitation of the Monte Carlo technique in the context of this protein energy landscape. So, the relationship between folding and the amino acid sequence was observed long back by Anfinsen. So, it is now known as Anfinsen's dogma, which says that the native structure is determined only by the protein's amino acid sequence. And he tested his hypothesis on the folding of ribonuclease A and got the Nobel Prize for this. However, it is long back. However, some limitations of his observations are noted. What he mentioned, if there is only the amino acid sequence which will tell what will be the native structure, then as if there is no effect of the environment. Say regarding the features or the driving forces that we mentioned, that what is the solvent, whether it is a water or lipid bilayer. Based upon that one, there will be some changes in the folding state. What will be the pH? What will be the concentration of the salt in the solvent? What will be the say temperature? So, based upon that one, the folded structure may change. So, one thing one example you can assume that the protein which say is assuming some stable state at room temperature, which you can consider as the 27 degree Celsius. If I keep on increasing the temperature, say if it goes beyond say 100 degree, 200 degree, whether it will assume the same structure? No, it is not possible. So, there are some limitations that it is not unique. So, one sequence can assume one structure if the environment changes. By environment, I used to mention that solvent, say pH of the solvent, then temperature, then concentration of the salt in that solvent. If it changes, then there will be some changes in the structure, and accordingly there may be some changes on the function. So, it is not unique. If I take the opposite way, I mean that if I consider one structure, whether corresponding to that structure there is only one sequence exists? No. We define protein design problem where I mentioned that corresponding to one protein structure there may exist more than one sequences. If more than one sequences exist, then you can consider that corresponding to multiple sequences you can map to one particular structure. Again, there exist multiple sequences who are very similar, but may assume to the same structure, but they are not same. Okay. Those sequences are generally called as the homologous. Another is the protein design problem, which says that corresponding to one protein structure there can be multiple protein sequences who are different. Okay. So, the stability may change again based upon the environment and kinetical accessibility. So, those things were not considered by the Einfinson. So, that is why some limitations we observed. Nevertheless, it was done long back and one of the classical work in this particular area. Kendall in 1997 published one very good paper in Nature Structural Biology. So, taking the exertion from there, he mentioned or gave one concept that in physics and biochemistry, an energy landscape is a mapping of all possible conformations of a molecular entity or the spatial positions of interacting molecules in a system and their corresponding energy levels. Typically gives free energy on a 2 or 3 dimensional Cartesian coordinate system, and also he gave this diagram. So, this diagram I taken from his paper, where you can see that the conformational space was mentioned here. This is my conformational space, and potential energy is in another dimension. So, we can assume that corresponding to one protein sequence you have generated all the possibilities, and say God is there, corresponding to each possibility God computed its energy function exactly what it is. Then if you plot them, then you will see something like this. Something like this means that if the potential energy decreases this way, okay, then it says that I am going to the native state, and that way you will see that on the surface, if I say this is my surface or conformational stage. So, here you will see most of the unfolded or loosely folded or just folding started this kind of situation structures. So, here, 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 here. Now, if you go inside, then it is taking more or less the correct shape or structure, and finally, you will go to this n, which is my native state. Now, this way if you go, and again I am assuming all the partially folded or intermediate, say starting from the protein sequence which is unfolded to the native state. When I am moving, I am generating all the possibilities. When I am generating all the possibilities, definitely they are following Ramachandran plot, okay, which means that there is a there is a they are following basically that phi psi angle of the Ramachandran plot. If I assume that one, then I will see there are some regions which are forbidden, means I cannot generate the conformation. That way some hilly regions are there inside, but overall it is looking like a funnel. It is looking like a funnel. So, that is why it is also called as the folding funnel, where these are my basically partially folded states, all are, and finally, when I will reach to this tip, that is my N, that is my native state. Okay. So, this is the hypothesis given by at the time. Next, if I take that particular energy landscape and project on a 2D, then you agree with me that it will be looking like something this. Okay. Here this is my potential energy and these are conformations. Perfect. This is my native state. This you can consider as partially folded state, or you can consider this as intermediate. Okay. So, this is my folding funnel. So, this intermediate state and this is my native state. Okay. Now, the first problem you should note, when say we are running Monte Carlo simulation technique, and why Monte Carlo simulation was not enough, but I have to go for Metropolis criteria. So, that I am going to demonstrate first. So, let us assume that you are started with some situation and it is your current situation, that is my ith instance. If you remember the Monte Carlo algorithm that we have discussed, corresponding to this there this basically ith iteration and current state is C. So, corresponding to this C, there is energy function E, say E of C. Now, it is a Monte Carlo simulation. So, randomly I am generating another move, that move let us assume is here. So, this move say I plus 1, and for that my conformation is C prime. So, energy will be E C prime, right. Now, theoretically if E C prime less than E C, then accept, that should be the situation theoretically. It is true if the surface is this folding funnel is something like this, which means it is gradually decreasing. So, that whenever you are generating some C prime, then the C prime will be either over C or below C, then it is fine, right. If it is the situation, this red contour is the folding funnel, then I can go by this, and that is my Monte Carlo simulation, and I am perfectly fine with it. Unfortunately, it is not the case. The case is here there is an intermediate state or partially folded state. If this is my partially folded state, then what will be the situation? Let me blow it up and draw here. So, I am having some state here and then it is going down. So, this I am calling this region as partially folded or say intermediate state. If it is, then what is my problem? You see, say this is my I. Okay. If this is my I, then I will get C here, E of C here. Now, I generate something here, say I plus 1, C prime is C prime. Okay. Now, if it is E C prime, then say that way I will reach to this position. Let us assume I have reached there and say that is my J. So, C J and E C J is my energy. After this one, if I make another random move and the random move is always in this region, then always the next move will have an energy function which is higher compared to C J. That way I will falsely assume that okay, I have reached to the native state or optimal state or the minimum state. Falsely I will assume that one, but that is not correct here. Okay. Because of this situation, because of this region, I am kind of trapped and falsely I am assuming that actually I have reached to the minima. However, it is not right. So, what will be my strategy here? That when I will reach to some region, then always, not always, I will accept when it is say this, but I will also accept when say E C prime is greater than E C. Sometimes I will accept this one also. Sometimes means how many times? For that I have generated one random number q equals to u 0 1 if q is basically, if q is basically greater than e to the power minus delta E by T, where T is the Boltzmann temperature and delta E is basically the difference between these two energy, then I will accept, otherwise I will not accept. That way I am ensuring that even if I will reach at this position, then I may take some region here as accepted, and if I accept that one, then my energy is not E C J now, it will be here. Next, when I will have another move, then there is a probability that I will pick somebody here, then it is with the minimum energy, this is minimum than this one. So, I will pick this one, that way I will keep on moving and I will go at this region. Is this clear? So, that is the reason why it is not M only, but metropolis criteria is also required for my purpose. Okay. Now, if I erase everything and say draw that folding funnel only, then this region is called as the local minima. This is also called as the local and if I reached here or say here, then that situation I will call as the locally trapped. So, I am trapped locally. For that I have to go for metropolis criteria. Okay. But sometimes that will not be enough also, that I will come later. When we are working with say computational technique, then basically I have a number of intermediate state. In computation I can call them as partially folded state or say decoys. So, such thing we already discussed in the context of protein complex modeling. In case of protein folding also there can be some decoys, decoys like this, decoys like this, this. Okay. Now, these decoys will have one individual energy. Say if without any loss of generality I can assume that okay. So, this is say E1, E2, E3, E4, E5, E6, E7, E8 and dot 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 dot. I do not know in the energy landscape or folding funnel where they resides. And also I mentioned about one situation where it will go to some amyloid fibrils and I mentioned that if it reaches there then it will not able to come back. So, that kind of situation is something when my folding funnel will be like this. Oops, sorry. If I assume some folding funnel like this, then this can be one native state and this will be these are the amyloid fibrils. The amyloid fibrils are looking like this, mostly they are with the beta sheets. They have pattern like beta sheets, and when it will reach there then you can see since the energy state is very less, even compared to the native state, then from here it is not possible to come back here, it is not possible. It is not possible to come back here. Okay. So, we have to be careful about that one also. Okay. Now, the external factors on protein trajectory. So, taking some of the examples or cases from the basically biological knowledge that we discussed on the last lecture, we can say modification of local minima by external forces can also induce modification of the folding trajectories, temperature, electric and or magnetic fields, molecular crowding, space constraints, those can happen. Okay. Now, the disruption of the native state. So, in this context it is better to discuss that point also. So, the native state or biologically functional forms may be disrupted for thermal instability that I mentioned, that say one protein is folded and having some function in the room temperature. Now, I am taking that protein and keep on heating, I mean I keep on increasing its temperature, then after say 100 or 200 degree etc., then some of its bond because of the thermal agitation will break and then it will start unfold. Okay. So, because of that one some unfolding may happen. High concentration of the solute, if I keep on changing the concentration of the solute, then also it may basically unfold or the native state may disrupt. Inadmissible pH. Okay. So, because of say high concentration of say charged high concentration of say acidic, if on the surface there are some basic amino acids, then they will try to go for the ionic interaction with the salt on the environment compared to its own interaction for the protein. So, that may disrupt the native state. Presence of chemical denaturants can do the same. Okay. Presence of chemical denaturants can do the same. Regarding this chemical denaturant, so most commonly used one is the urea. So, if you add urea to protein, mostly it will it will denature or it will disrupt the native state, but you have to remember that not all disruption is reversible in nature. So, mostly if the native state is disrupted, then it will not fold or it will not take the previous native state. Denature, refolding and aggregates. So, a fully denatured protein lack both tertiary and secondary structure and exist as a so called random coil. Mostly denaturation is irreversible, that I have mentioned just now. Chaperones or heat shock proteins, in short this is a very famous protein actually, this is called as the HSP proteins, protect against the denaturing. So, how? Interesting, I mentioned on the last lecture also. In some situations some misfolded proteins are unfold for a second chance to refold properly. This function is crucial to prevent risk of precipitation into insoluble amorphous aggregates. But if given the second chance to refold properly also fails, then perhaps it will go to some precipitation or insoluble amorphous aggregates, which may lead to some disease condition, mostly the neurodegenerative diseases kind of thing. Okay. Incorrect fold and neurodegenerative disease. Aggregates or misfolded proteins accompany illness. So, these are the list of the illness, these are not only, there may be several others. So, researchers are working on this one, so kind of Jacobi's disease, bovine spongiform, encephalopathy, mad cow disease, then amyloid related illness such as Alzheimer's disease, familial amyloid cardiomyopathy or polyneuropathy, intracytoplasmic aggregation disease such as Huntington's and Parkinson's disease, antitrypsin associated emphysema, cystic fibrosis, lysosomal storage disorder. So, this kind of situation may happen if a protein is not correctly folded or it assumes some incorrect folding state. Again, if it goes for incorrect folding, then it gets mostly another chance to open and refold, but even if it fails, then it will lead to some incorrectly folded state. And as I mentioned, it is not possible for the immune system to identify all the miscorrect or misfolded or incorrectly folded state. If it cannot identify that one, then that will definitely lead to some disease condition, something like this. Now, regarding the folding funnel I mentioned, but few remains. So, let us assume that okay, this is my native state, make it simple now, do not incorporate the kinetically controlled or amyloid fibril situation, this is my native state. Now, even then also you will find that so many local minimas are there. So, these two local minima may not be very severe, but these are very severe because they are close to native state. Once I will reach there, then it will be problem. So, only Monte Carlo simulation may not give me the correct solution or suggestion for a protein whose folding funnel may look like this. If that is the situation, then what shall I do? And before that one, perhaps you noted during the Monte Carlo simulation technique. Okay. So, if say let me pick say dark color. If my situation is here, then I can randomly move, that is fine, but say it is Monte Carlo simulation. So, I am starting with some random state, but if my random state starts from here, say pink color, and it always generates states within this region only, then again incorrectly you will lead to one situation which is called as the locally trapped situation. So, we will be trapped locally in this locality, you will not able to reach to the native state. If it is the situation, then what to do? But problem probably you identified that starts with the initial seed or random. So, it is here. Now, it can be here also. If it is here, then also it will be the same thing, or it can be here also. If it is here, then I will bingo, I will get the native state by minimizing that one. The problem in this case is that you have the correct energy function, assuming the perfect energy function, then also what is your seed or from where you are starting, based upon that one a lot of things may change. Okay. So, starting with one seed may not be enough. If not, then what if I start with say multiple seed values with a hope that if all the seed values, say random seed values, are random in nature, which means that there is no correlation between the initial seed values also, then there is a probability that once a seed value, let me take the green, will be here, another 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 here. That way if I go, and during the process or iterations or simulation of the Monte Carlo simulation, if it is then during the simulation process, if I combine the results or compare the results, then I can able to understand, okay, so who is here can reach up to here, who is here can reach up to here, who is here can reach up to here, others can go somewhere else. Now, comparing this and this, I can able to understand that what is the optimal energy. Okay. So, based upon that one, another extension of our Monte Carlo with Metropolis criteria method will be modified, that is called as the replica exchange, REMC, that we will discuss in the next lecture. Okay. So, now let me conclude this with the experimental techniques which are there to determine the structure of the protein. It is X-ray crystallography, protein nuclear magnetic resonance spectroscopy, circular dichroism, dual polarization interferometry, vibrational circular dichroism of protein, studies of folding with high time resolution, proteolysis, optical tweezers, etc. Now, you should remember that these techniques are not required for our course purpose, but I mentioned this one, so that since you are working with the protein, so you should know at least that what are the different experimental techniques are there, or when you talk to biologists or experimentalists regarding the development of the algorithm or to understand their problem, then you should know that these techniques exist but we are not going to discuss these techniques for our course. Okay. That is it. Thank you very much.
Swayam Prabha. Digital India. Educated India.
Vortex coloring and upper bounds. Preface. Recap of previous lecture. We have discussed the network flows, maximum network flow, F augmenting path, Ford-Fulkerson labeling algorithm, max flow, min-cut theorem. Content of this lecture. This lecture we will discuss graph coloring, that is the vortex coloring and its upper bound. Vortex coloring